He said that is non-stop. Right now. So I'll just add the references. Yeah, they're already, he already put them in there. I just, literally we just I think, like, I'm not, don't take this, but when I did it, literally you write one and there's like dash and then the source, like the cited source at the bottom. Yeah. No, you just leave the numbers. So like this one's 2, so whatever source you use for 2, you'll be like, well obviously you do 1 first, but you'll be like 1. No, this is really cool. I just don't understand. We have a plot of land for you to take in camp. And I said, that's just not happening for me. I'm sorry. Are we starting a new lecture? Or just finishing the class?
Yeah, basically finishing what we left off with, but then also there was a couple of things I wanted to go through the beginning of today's lecture that are additional, like forum sensing and stuff, but it's just several slides, it won't be too much.
Okay, I just didn't know if it was on the same slide, sure.
Yeah, so there's overlap in the end of lecture 9 slides and then the beginning of lecture 10. I just, yeah, left it there.
No, exactly. You should be Kelly. You guys are fine. I love your board. Yeah. How do you never been out of Detroit?
I only know from twice.
Oh, yeah, that's one. Let's go to last,
I go
right?
Columbus to Chicago. And then on the way back, I went to Texas.
Sometimes we one time I was to Georgia to get my. Or went to Georgia to go to Mexico. Nobody's there.
All right. Good morning, everybody. Happy Monday. So today, like I said last week, we're gonna just continue on the rest of what was in lecture 9, and then we're just gonna go through a little bit more content on quorum sensing. So should be a shorter lecture just to wrap everything up, this entire unit. This will be your last lecture for the second exam. So your second exam will be after break, so you guys will have a large chunk of time, so that could be good because you guys could potentially be able to study more, but then it also could be bad, you guys could forget the material, so just try to stay on top of it. And so, like I said last week, because bacteria are constantly going into these different environmental conditions, the bacteria have to know when they need to alter their physiology and regulate their gene expression. So today we're going to talk a little bit more about some operon control, and then we'll get into quorum sensing and how bacteria communicate. They have these languages in which they communicate just like we do. So we talked about the lac operon. The lac operon is the lactose operon, and it's used as the model usually to teach how operons work. Well, bacteria have many other ways to regulate their genes, and so some proteins have these dual regulatory functions. And so what that means is that they have literally 2 regulatory functions. So the 2 we talked about are activation and repression. Some proteins can do both themselves. And so they can serve as both an activator and a repressor depending on the environment and switching. And we talked about how in bacteria there's coupling of both transcription and translation, and so this also becomes important when regulating gene expression because they can use this mechanism called attenuation that we'll go through a little bit briefly. But essentially what attenuation means is that it basically involves utilizing ribosomes at the translation part, and the ribosome is this sensor that can send chemical signals that can then express or affect the expression of the mRNA transcript being made. So I'll talk a little bit more about that as well. Any questions on anything from last week on the lac operon? Okay. So we're going to talk about something that will have both the regulatory protein has dual activities, so it can be the activator and repressor. And the classic example here is this arabinose operon. And so arabinose is just an important carbon source, and so its genes will convert in the arabinose operon, the genes will convert arabinose into xylulose P which is an intermediate and an important biosynthetic pathway. You don't really need to know that. All you need to know is that arabinose is an important carbon source, and usually we're breaking it down using this operon. And the major regulator protein that acts as both the repressor and the activator is ARA-C. And so it's a dimer, and it can either repress or activate gene expression, and it's all dependent on whether arabinose is present or not. So when arabinose is absent, the ARA-C dimer is elongated, and I'll show you a picture of that, but essentially when arabinose is absent, this ARA-C will repress the expression of the genes that break down arabinose. And if you think about it, why bother express the genes that will break down arabinose if arabinose is not there, right? Kind of similar, common sense to what we've been talking about. When arabinose is there, then the ARA-C regulator can stimulate then the binding of the RNA polymerase to the promoter and then transcription. So really, this is the same protein, and it's this dimer, but it's a conformational change that makes it either a repressor or an activator, and that's all dependent on whether arabinose is present, the thing that it wants to break down. So this is the model for the ARA-C regulation. It's also sometimes called ARA-BAD operon, just arabinose, and then I think the D is like the dimer part of it. And that regulator is ARA-C. So here we're looking on this side where ARA-C is there, but there is no arabinose. And so you can see this ARA-C is this dimer, so these two proteins, and it's in this elongated form. And so once it's in this form, then basically it will repress the expression of this operon. And the way that it does this is this ARA-C and this conformational change can then bind over here, and then what you need to know is that it creates, because of the conformation that it's in, it's kind of like, this is the DNA, and it's kind of like pushing the DNA like away from each other to create this loop, and so it stops transcription. Does that make sense? So this DNA loop blocks transcription, and DNA loops technically do that. And so when arabinose is present, though, this dimer structure compacts, and so these binding domains actually are immediately adjacent to one another. And so this conformation allows this to bind the operator region, and so you no longer get this DNA loop that represses, and so then you can activate the RNA polymerase and ultimately allow for the transcription of the arabinose operon. So it's the same protein, ARA-C, that's acting as a repressor or an activator, but it's all dependent on whether this ligand or arabinose is present or absent, and that initiates a conformational change. In the elongated form, it's going to create this DNA repression loop and repress transcription. In this more dimerized conformation, then it can bind the DNA region and promote transcription. Make sense? So we've talked a lot about operons for catabolism. Catabolism means breaking things down. The way that I remember this from literally high school chem is catabolism. I think a cat likes to break things. So I don't know, that's just my way of thinking about like breaking things down. And so catabolism is the breaking down of carbon sources. And so today we're going to talk a little bit more about regulating anabolic pathways. Anabolic pathways are the opposite, where you're building things up. And so the regulation of catabolic versus anabolic pathways is fundamentally different. And the reason is because when you're making something, you're building something up. For instance, the cell needs to make amino acids, right? So we're going to talk about this in the context of amino acids. And so say we have an amino acid like tryptophan. You start with very little, and then you have to make more, and then you have a lot in the end. So it's different from catabolism. And so eventually you'll have enough tryptophan in the cell, this amino acid, and so then it has to turn the anabolic operon off. And so basically to repress that anabolic pathway, so stopping the synthesis of tryptophan, for example, it's pretty different from repressing a degradative system or catabolic system. And so basically this is typically regulated by repressors called these inactive aporepressors. And so in this situation, in this mechanism, normally these aporepressors only bind when they're and are able to repress when there is a co-repressor. And this should make sense because the co-repressor is the end product of the biosynthetic pathway. So if the end product is the amino acid tryptophan, then once you have enough tryptophan in the cell, that will act as a co-repressor, binds this inactive aporepressor to activate it, and then this complex can then bind the operator sequence and block transcription, block the RNA polymerase. Does that make sense? So I know it makes sense as we kind of talk through it, but I really want you guys to keep these things, straight in your head, especially for an exam. If I ask a question about an anabolic pathway, you have to kind of think back that, we're talking about the tryptophan operon, for instance, or when I talk about catabolic pathways, there's the lac operon, there's activators, repressors, so many different things that are involved in it. And then there was the catabolic one where I just talked about that rhabinose operon, where that one is different from the lac operon in that it has the same regulatory protein having the dual function. So try to keep those things in mind. And so this is called a negative feedback mechanism, essentially, because when you have too much of the thing that you need, it has to negatively, it has this negative feedback mechanism. You're using that thing that you're making to repress the operon at the end. So let's kind of go through this a little bit more again with some pictures. So the TRP operon, or the tryptophan operon, is the biosynthetic operon, encoding enzymes that are involved in tryptophan production. So not breakdown, but production. And so when the tryptophan levels exceed the cell's needs, then that excess tryptophan, which ends up acting as the co-repressor, binds to the inactive aporepressor, which here is this TRPR. And the R basically will remind you that this will be a repressor when it's active. So basically, this complex will then convert into an active repressor, and so now it can bind to this operator sequence. So here we're looking at the TRP repressor, TRPR, and so it's inactive. And so here's the region where it would want to bind to then repress the transcription of these genes. And so once the co-repressor binds, so tryptophan will bind to the aporepressor, you get this holo repressor. And so then that can bind to the operator and greatly reduce transcription. Okay, and so repression is not the only mode of regulation of this TRP operon, and so I briefly talked a little bit about attenuation. A lot of amino acid biosynthesis operons use this transcriptional attenuation. There are so many amino acids being formed inside of the cell, and so basically what this means is that the cell is using the ribosome as a sensor of amino acid levels in the cell, and so remember that for a ribosome to work, it needs to have these charged tRNAs, and the tRNAs won't be charged efficiently if you have low levels of that particular amino acid. So you need a lot of that amino acid in order to charge the tRNA and in order for that tRNA to take that amino acid to the ribosome. So it's all connected. And so basically, eventually, you'll get ribosomes that are getting tRNAs coming in that are not charged. So now the ribosome can be used as a sensor for what amino acids the cell is depleted for. And so this plays a role in this whole concept of attenuation. So think of it as a transcriptional control mechanism. Basically, Another way to put it is the ability, and you don't have to know any of this, this is kind of just for you guys to, you don't have to know anything about the different stem loops that are forming, but essentially what you should, try to understand here is that the ability of the cell to be able to translate part of the mRNA determines whether the transcribing RNA polymerase will continue to make that mRNA transcript upstream. So, I guess, does that make sense? A little bit? Okay, so basically there's translation happening in a leader peptide, and it can sense how much of an amino acid is in the environment, and if it senses that there's a lot of that amino acid and we don't need to keep making more, then that translation of that leader peptide can then influence what's happening beforehand downstream and stop the transcription of an mRNA. So it's like a transcriptional control mechanism. This all goes back to the fact that transcription and translation are coupled in the cell, so they all can affect each other. And yeah, so basically the reason for this is that because attenuation will halt transcription that's still occurring in progress, it provides this quicker response to changing amino acid levels compared to just simply repressing that transcription that we talked about earlier. So attenuation basically will halt transcription in progress. So let's kind of talk through this a little bit together. So here we have high tryptophan levels, so the ribosome is right here, and so it's basically translating and it's doing its thing, and it has to encounter the stop codon, and so basically the ribosome stops and it's covering certain regions, and you have the stem loop forming, and so basically this loop binds the RNA polymerase and causes the RNA polymerase to then release and stop making this mRNA transcript because there's a lot of tryptophan in there. Whereas when you have low tryptophan levels, the ribosome initiates translation of, the leader mRNA, and so there's scarce tRNA. Basically, the ribosome is getting these tRNAs that is telling it like, oh, these tRNA aren't charged, and so the ribosome then can stall and then basically attenuates this whole situation, the whole attenuation of the transcription. And so basically that results in the stem loop can't form, and so the polymerase is able to then transcribe the trip operon. So basically ribosome senses that there are low tryptophan levels because the charged tRNAs aren't charged coming up to it, and so it tells the transcription downstream, or the RNA polymerase downstream to continue transcribing that mRNA transcript. Okay. So that's all you have to know for operons, that's in the previous lecture. And so I'm going to talk a little bit about quorum sensing, which is just another type of gene regulation, but where the bacteria are able to signal, and it's not just occurring intracellularly. The bacteria are allowed to signal for basically by communicating with one another. So it allows a population of bacteria to communicate with one another and work together. So bacterial cells work together only at high densities. And so this was discovered in this bacteria called Vibrio fischeri. I also call it Vibrio fischeri. It's actually really, really cool. Basically, it's this bioluminescent bacteria that colonizes the light organ of the Hawaiian bobtail squid. So basically, this is the bobtail squid here. And so when you grow Vibrio fischeri on a plate, it looks just like this, kind of like what E. coli looks like. But then if you turn the lights off, it glows. It has this bioluminescence property. And so basically, one of the ways that this was discovered, or the way it was discovered, was basically this bacteria in like water would be like, basically it would go into the squid and allow the squid to luminesce, and so that was then beneficial for the squid to be able to like do things for itself and find prey and things like that. So the reason why it can do this is because it expresses this bioluminescent molecule. But it doesn't express that bioluminescent molecule all the time. If I give you just one cell, the Vibrio fischeri, you're not going to see this at all. And so that's the important thing here. It doesn't make sense for a single cell to turn the flashlight on, essentially. You won't really see anything. So instead, these cells are able to release these molecules that we call autoinducers. And these autoinducers measure the concentration of that autoinducer in that entire environment in order to determine that they're at a high enough cell density to then turn these gene sets on, and in this case, these are the genes for bioluminescence. So here we have the cell, individual cell, and this is the autoinducer. This is this chemical that it's releasing. And so nothing happens. But then when you have enough of these, then the bacteria know, oh, okay, well, there's enough of a concentration of these autoinducers around, so there's a lot of bacteria around me that are going to do the same thing as me, so it's time to all turn our light on. It's a group behavior. And so over here, I'm just showing you over time. Arbitrary units is on the y-axis, but essentially in red here, I'm showing you the biomass, so just the growth of that bacteria, and how it grows over time. And so you can see here that this in blue is the bioluminescence, so you can see that usually it's like growing and it's fine, and then the bioluminescence is low, so you have just like some cells, but then once there's a particular density of the biomass, enough cells in that area, in that population, then you have this, it shoots up and they all start to bioluminesce. So it's very specific for like a certain cell density that they have to reach. That cell density corresponds to the amount of the autoinducer that's there. It really only works like that in teens, essentially. Okay, so the autoinducer is the molecule secreted from the cell, and it has the ability to also re-enter the cell. So it's at certain extracellular concentrations, the secreted autoinducer will re-enter the cell. So this basically, this autoinducer has to bind to a regulatory molecule, so it's another, regulatory transcriptional type of scenario here. And so in this case of Vibrio fischeri, that regulatory molecule is LuxR. So this LuxR plus the autoinducer complex can activate transcription of the genes that will allow it to bioluminesce. So here we have this LuxR protein, and so normally it's just repressing, but then you have these cells basically making all of these autoinducers, so that's the LuxI protein, this acyl homoserine lactone autoinducer. You don't have to know any of these structures, but you should know that the cell, this is the cell, it's making this autoinducer, and so the autoinducer starts to accumulate and accumulate and accumulate outside of the cell, and then this autoinducer can then, when it reaches those really high extracellular concentrations, it can then come back into the cell. And so at that threshold concentration, this autoinducer binds this LuxR repressor, and so binding of the repressor relieves the repressor off of the mRNA transcript, and so it kicks off the repressor from the operon, and you can then have transcription of those bioluminescence genes. Does that make sense? So the autoinducer binding the LuxR relieves the repression because it's initially repressing those genes that will cause bioluminescence. And so once you have that high concentration and they want to start to express their bioluminescence genes, then you have to remove the repressor that's stopping the expression of those genes. And that's all done by this autoinducer. So this chemical is what drives that. Does that make sense? Any questions? Okay, so let's watch a little video, just reiterating the concept.
Off the coast of Hawaii lives a cunning nocturnal hunter with a brilliant trick up its sleeve, or rather its mantle. Meet the Hawaiian bobtail squid. After spending the day sleeping buried in the sand, come nightfall, the little squid emerges in search of a meal. To hide itself from predators and prey alike, the squid has its very own invisibility cloak, a colony of a glowing bacterium called Vibrio fischeri, which it houses in a special organ in its mantle. In an ingenious form of camouflage known as counter-illumination, the squid uses its glowing bacteria to mimic moonlight, hiding its shadow from any creatures looking for an easy meal. Moving through the depths, the squid uses its ink sac to adjust the intensity of its living torch to match the strength of the moonlight. Also handy if a cloud suddenly passes in front of the moon. Vibrio fischeri glow because they have genes for producing light using a simple chemical reaction. Scientists growing Vibrio fischeri in the lab notice something quite interesting. The bacteria only switch on their light when they have reached a critical density. This makes perfect sense. There's no point going to all the trouble of making light if it isn't bright enough to be seen. But how do they do it? Each bacterium produces a chemical signal called the autoinducer. The more bacteria there are, the more autoinducer there will be. If those bacteria are growing in a confined space, like a flask, or the light organ of a squid, the amount of autoinducer will start to build up. Once the autoinducer reaches a critical concentration, it triggers the bacteria to switch on the genes for producing light. This chemical form of communication is called quorum sensing. It turns out that many species of bacteria use quorum sensing to coordinate their behavior, from sex to swimming to causing disease in plants, animals, and humans. Scientists hope that one day quorum sensing may be exploited to turn the tables on some of our deadliest foes. And if they do, it may well be thanks to that little nocturnal hunter, the Hawaiian bobtail squid.
Okay. My first time watching that video, so I'm glad I taught that appropriately. So that was just a good description of that. And so one of the pioneers of this field is Dr. Bonnie Bassler. She is actually probably one of my biggest inspirations. So when she was discovering this whole thing, I was in, my undergrad and grad school and just kind of following up with her work in this. And then she actually has this really, really cool TED Talk. You can find it on YouTube if you just look up TED Talk, Bonnie Bassler, quorum sensing. And she explains all of this, and it's the coolest thing ever. Her work is so cool. And she explains it in a way where any audience can understand this. But essentially, yeah, she basically pioneered this field. She's in your textbooks now and everything. And so she says that she showed it, and her group showed that bacteria use quorum sensing both for universal communication with other bacteria, as well as secret conversations in the same bacterial populations or species. And so basically, they, you don't have to know all of these, but these are all different types of bacteria that form these autoinducers, and you can see that all of the autoinducers on one side look the same, but then there are slight differences on the other side. But essentially, what they're doing is they're counting their siblings in a way. And so the autoinducers you can think of as a language, and they can, these languages can be for different bacteria that are related. Autoinducers of different bacteria are related but different. So these different bacteria have different autoinducers, but they're still related because they have similar structures on the left. And so this particular example of having different autoinducers for different bacteria allows for species specificity. So Vibrio fischeri can then count their siblings by just releasing this autoinducer, and this Pseudomonas aeruginosa count the other Pseudomonas aeruginosa in the area by basically releasing this autoinducer. And so this kind of helps them decide when to attack, and so this is an example of intra-species communication. But like I said, they can also have auto-inducer molecules that more than one species of bacteria can recognize, and so it's more of a universal language as well. So it could go either way, depending on what they want to do. So why are bacteria doing this? It's not just for light and this bioluminescence. So there are, quorum sensing is important in pathogenesis, and that video kind of alluded to that, where scientists today can kind of target some of these things that bacteria can do in order to then maybe make antibiotics and things like that. So there are many pathogens, many bacteria that are pathogenic that use quorum sensing to control virulence genes. So it's not just for producing light. And virulence proteins could be proteases and enzymes that degrade things. And so these genes are not produced until the cell density is really high and it can overwhelm the host. So similarly to just having the autoinducer expression for light. So it's always going to need the cell density to be at an optimal amount for it to be able to express the genes that this is controlling. So one of the recent questions that the Bachelor group and others are asking is, but bacteria live in these very diverse communities, so do different bacteria also talk with each other? And so that was kind of what I was alluding to with, the universal language. There is also interspecies communication. So inter means between species, and so basically universal autoinducers. There are universal autoinducers that several bacterial different species can recognize and respond to, and so this is kind of a new target for antibiotics. Basically, can we trick the bacteria into thinking that they're always in the minority, so they never express those virulence genes? So what I mean by that is interspecies quorum sensing basically tells the bacteria whether they're in the majority or minority. And remember, they only express those genes, whether it's bioluminescence or the virulence genes for their pathogenesis, they only express when they are in the majority. So they have to ask, like, oh, should I put up a fight or should I lay low? So they need to be in the majority for this quorum sensing to happen. And so basically, can we prevent bacteria from talking and listening to each other by targeting these quorum sensing things for specific and broad spectrum antibiotics? So, can we basically trick them into thinking that they're just still in the minority by, attacking that concept that they have? Does that make sense?
So they need to be in the majority.
Yeah, exactly. And being in the majority means that you are at that very high cell density. Okay. So far we focused largely on these individual gene circuits, but there are several behaviors of organisms that require these integrated gene circuits. So they're more complex decision making. So we're going to kind of, veer away a little bit from operons and quorum sensing and just talk a little bit about this concept called chemotaxis. And so it's just important to kind of go through this because we are going to be doing a little bit of, we're going to try to do a motility experiment tomorrow where we look at organisms moving, and that's in part due to their flagella and being chemotactic. So basically, a bacteria has to know when it should swim and in what direction it's swimming towards, either a chemoattractant or away from a repellent. So if we give it antibiotics, it's going to want to swim away from that to repellent, and that's really not that random. It has to know when it should swim and when it shouldn't. They have these flagella, and this requires a building of multiple switches with feedback controls. So one of these examples is chemotaxis. So yeah, many bacterial genes are regulated by this hierarchy of regulators, so there's more than one, and they form these integrated circuits. And they basically can do these gene switches to regulate their decision making because they can be moving towards something and then run into something that is a repellent and then have to like switch and move away, and so it's very regulated. And it involves mostly these single switch mechanisms. So bacteria that use flagella move, and so they don't just simply dash about hoping that they swim into a favorable condition. There's this chemotaxis that allows the ability of the organism to sense chemical gradients and modify its motility in response. This is different from quorum sensing, and there's chemical gradients that they are sensing in both cases, but in chemotaxis, it's not a population of cells that have to reach a high density like it is in quorum sensing. In chemotaxis, it's that single organism being able to sense something that's either an attractant or a repellent, and so it modifies its motility in response by using this flagellar motor. So it's not necessarily random. The direction of this flagellar motor rotation determines the type of movement. So if it's going in the counterclockwise rotation, it's smoothly swimming, it's directed and smooth, and then if it's going in this counterclockwise rotation, they can tumble. And to give you a little bit more of an example, so this is a paratrichous flagella. So I talked about this very early on, where paratrichous just means that the flagella are randomly positioned across the entire length of the cell. They're not just coming out from the back, it's really kind of all over. And so when it's in the counterclockwise rotation, all of these flagella kind of are oriented in a bundle behind one pole of the cell. So basically, when the cell veers away from the attractant, there are receptors that allow the flagella to switch rotations as well. So the counterclockwise allows it to swim toward an attractant, and then if it needs to like veer away from the attractant a little bit for some reason, maybe there's a repellent or a decrease in the attractant concentration, that can cause a switch to the clockwise rotation. And so in the clockwise rotation, then you start to see this tumbling where you have these flagella kind of not really bundled up at one pole anymore. They're kind of all over the place, and they cause this bacteria to start tumbling. And they're all rotating in a different direction, so it kind of doesn't know where to go. It's like canoeing with a friend and you're both going like the opposite directions, and it's kind of like, you're kind of stuck in that sense. That's kind of what I like to think of it. But so these are these alternating runs and tumbles that you can have, and it's important to note that like bacteria kind of switch through those. There's really a lot of alternating, like running smoothly and then tumbling a little bit. And so this pattern of movement is this biased random walk, that's what we call it, even though it's not a random, like the counterclockwise rotation is not random, clockwise rotation isn't random, but this net movement is kind of random. And the way that I think about this, and it's a good analogy that I've been given before, is think about the net movement is like going home after the bar, if you're like walking home, and so your net movement, you're going from the bar to your home, but then you're kind of like, stumbling home, and maybe you stop at 7-Eleven for a bit, or maybe you fall in the ditch for a bit, and then like get up and like keep moving forward, or like, maybe you like run into a friend or something and like talk for a little, or you need some pizza real quick, something. So, but eventually you get home. So that's kind of this biased random walk. That's kind of how I like to think about it. But yeah, so that's really what you guys need to know. There is this whole, E. coli has flagella, like I've said, and they can move, and that's one of the things we'll try to look at tomorrow in lab. But there's this whole signaling pathway that allows it to do this. I have this up here. If you're interested, you could read about it more in the textbook, but you don't have to know this necessarily for the exam, but if you are interested, it's just another signaling pathway very similar to some of the things we've been talking about. I just didn't want to bombard you with more, but that's really all that you would need to know for Unit 2. Are there any questions? Okay, so I have worksheet 4 here, which is going to summarize some of the concepts for lectures 9 and 10. So it's all really chapter 10. And so Yeah, you guys can just pick up a worksheet. This time I didn't give you enough space to write on this, so feel free to print it out. It's on Blackboard if you want, and type it out, or just use a different paper, but pick one up before you head out.